Biomolecules Part I. In the living organisms, there is a wide diversity in our biosphere. Now, in our minds, a question that arises is, are all living organisms made of the same chemicals, that is, compounds and elements? In chemistry, you have learned how elemental analysis is performed. Such an analysis, if performed on an animal tissue, plant tissue, or a microbial paste, we get a list of elements like hydrogen, carbon, oxygen, and several others and their respective content per unit mass of a living tissue. We obtain a similar list if we perform the same analysis on a piece of Earth's crust as an example of non-living matter. Between the two lists, what are the differences? No such differences in absolute terms could be made out. All the elements present in a sample of living tissue are present in a sample of Earth's crust. A closer examination, however, reveals that with respect to other elements, the relative abundance of carbon and hydrogen is higher in any living organism than in Earth's crust. How to analyze chemical composition? In the same way, we can continue asking in living organisms what type of organic compounds are found. How does one go about finding the answer? One has to perform a chemical analysis to get an answer. Any living tissue can be taken and using a mortar and pestle, the tissue is ground in trichloroacetic acid. A thick slurry is obtained. We would obtain two fractions if we were to strain this through a cheesecloth or cotton. One is called the filtrate or the acid soluble pool more technically, and the second the acid insoluble fraction or the retentate. In the acid soluble pool, thousands of organic compounds have been found by the scientists. It will suffice here to say that the compounds are extracted, then the extract is subjected to various separation techniques till one has separated a compound from all other compounds. In other words, a compound is isolated and purified. When applied to the compound, analytical techniques give us an idea of the probable structure of the compound and the molecular formula. From the living tissues, all the carbon compounds that we can get are called biomolecules. Living organisms, however, have also got the compounds and inorganic elements in them. How do we know this? A slightly destructive but different experiment has to be done. A small amount of a living tissue is weighed and dried. All the water evaporates from the living tissue. The remaining material gives dry weight. All the carbon compounds are oxidized to gaseous form if the tissue is fully burnt and are removed. The remaining matter is called ash. Some inorganic elements like calcium, magnesium, etc. is present in this ash. Also seen in the acid-soluble fraction are the inorganic compounds like sulfate, phosphate, etc. Therefore, elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. are given by the elemental analysis, while analysis for compounds in living tissues give an idea of the kind of organic and inorganic constituents present in it. One can identify functional groups like aldehydes, ketones, aromatic compounds, etc. from the point of view of chemistry. We shall classify them into amino acids, nucleotide bases, fatty acids, etc. from the biological point of view. As substituents on the same carbon, that is the alpha carbon, amino acids contain an amino group and an acidic group and are organic compounds. Hence, they are called alpha amino acids and they are substituted methanes. Occupying the 4 valency position, there are 4 substituent groups and these are hydrogen, carboxyl groups, amino group and a variable group. They are designated as R group. There are many amino acids based on the nature of R group. Those which occur in proteins, however, are only of 21 types. In these proteinaceous amino acids, the R group could be a hydrogen, a methyl group, hydroxymethyl, etc. The physical and chemical properties of amino acids are essentially of amino, carboxyl, and the R functional group. On the basis of the number of amino and carboxyl groups, there are acidic, basic, and neutral amino acids. Similarly, aromatic amino acids are also present, example tyrosine, phenylalanine, and tryptophan. The ionizable nature of NH2 and COOH groups is one particular property of amino acids. Hence, the structure of amino acids changes in solution of different pH. Generally, lipids are water insoluble and they could be simple fatty acids. Attached to an R group, a fatty acid has a carboxyl group. A methyl or ethyl or higher number of CH2 groups could be the R group. For example, including carboxyl carbon, palmitic acid has 16 carbons. In arachidonic acid, including the carboxyl carbon, it has 20 carbon atoms. Fatty acids can be saturated or unsaturated. Glycerol, which is trihydroxypropane, is another simple lipid. Both glycerol and fatty acids are present in many lipids. Here, esterified with glycerol are found the fatty acids and they can then be monoglycerides, diglycerides and triglycerides. Based on melting point, these are also called fats and oils. Oils remain as oils in winter as the oils have lower melting point. From the market, can you identify a fat? Phosphorus and a phosphorylated organic compound are present in some lipids. These are phospholipids. They are found in cell membrane, example lecithin. Especially some tissues have lipids with more complex structures, example the neural tissues. A number of carbon compounds are present in living organisms in which heterocyclic rings can be found. A few of these are nitrogen bases, adenine, guanine, cytosine, uracil, and thymine. These are called nucleosides when found attached to a sugar. They are called nucleotides if a phosphate group is also found esterified to the sugar. The nucleosides are adenosine, guanosine, thymidine, uridine, and cytidine. Nucleotides are adenylic acid, thymidyllic acid, guanylyl acid, uridylic acid, and cytidylic acid. DNA and RNA, the nucleic acids have nucleotides only. The DNA and RNA function as genetic material. Primary and secondary metabolites. Isolating thousands of compounds, big and small, some living organisms is the most exciting aspect that chemistry deals with, if possible, synthesizing them and determining their structure. If a list of biomolecules were made, then the list would have thousands of organic compounds including amino acids, sugars, etc. These biomolecules can be called as metabolites. These are called primary metabolites. However, when plant, fungal, and microbial cells are analyzed other than these primary metabolites, one would see thousands of compounds, example alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. While identifiable functions are present in primary metabolites and in normal physiological processes, they play known roles at the moment. The role or functions of all the secondary metabolites in host organism is not understood. However, to human welfare, many of them are useful. Some of the secondary metabolites do have ecological importance. biomacromolecules. To all those compounds found in the acid-soluble pool, there is one common feature. Approximately molecular weights ranging from 18 to around 800 Daltons are present in them. Only four types of organic compounds are present in the acid-insoluble fraction, that is lipids, proteins, nucleic acids, and polysaccharides. With the exception of lipids, these classes of compounds have molecular weights in the range of 10,000 Daltons and above. For this very reason, chemical compounds, that is biomolecules present in living organisms, are of two types. Those that have molecular weights less than 1000 Dalton and are referred to as micromolecules or biomolecules, while those that are found in the acid insoluble fraction are called macromolecules or biomacromolecules. With the exception of lipids, the molecules in the insoluble fraction are polymeric substances. Then why do lipids come under acid insoluble fraction, that is macromolecular fraction, when their molecular weights do not exceed 800 Daltons? Lipids are small molecular weight compounds and are present not only as such, but they are also arranged into cell membrane and other membranes. We are disrupting the cell structure when we grind a tissue. When the cell membrane and other membranes are broken into pieces, they form vesicles which are water insoluble. Therefore, in the form of vesicles, these membrane fragments get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid-soluble pool roughly represents the cytoplasmic composition. From cytoplasm, the organelles, the micromolecules, become the acid-insoluble fraction. The entire chemical composition of living tissues or organisms are together represented by them. In summary, we observe that water is the most abundant chemical in living organisms if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise. Proteins. Proteins are polypeptides. Linked by peptide bonds are a linear chains of amino acids. Each protein is a polymer of amino acids. A protein is a heteropolymer and not a homopolymer, as there are 21 types of amino acids. Only one type of monomer repeating n number of times is present in a homopolymer. The source of essential amino acids are the dietary proteins. Hence, amino acids can be non-essential or essential. The non-essential amino acids are those which our body can make, while we get essential amino acids through our diet or food. Many functions in living organisms are carried out by proteins. Some are enzymes, some fight infectious organisms, some are hormones, some transport nutrients across cell membrane, etc. In the animal world, collagen is the most abundant protein, and the most abundant protein in the whole of the biosphere is ribulose bisphosphate carboxylase oxygenase or Rubisco. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.